Now let's go on to the next type of cell division, which is meiosis. Meiosis occurs in eukaryotic organisms that reproduce sexually. This includes plants and animals. Meiosis is a two-part cell division process that produces sex cells with one-half the number of chromosomes as the parent cell. There are two stages or phases of meiosis: meiosis 1 and meiosis 2. At the end of meiotic process, 4 daughter cells are produced. However, before a dividing cell enters meiosis, it undergoes a period of growth called interphase. The interphase is divided into 3 phases. The first one is G1 phase. This is considered as the longest phase of interphase. In this phase, the cell increases in mass in preparation for cell division. Duration of this phase is varied depending on the kind of cell, its location, and function. In embryonic tissues, cell increases in volume by imbibing water and nutrients. The second phase is the S phase. This is considered as the most critical and significant phase because this is the period during which DNA is synthesized. Note that S represents synthesis. The last phase in interphase is G2 phase. This is where the period between the completion of DNA synthesis and the start of active mitosis with many internal chemicals changes in the cell. Cell synthesizes proteins and continues to increase in size. The duration between S and G2 is phases variable depending on the cell, but both are shorter than G1 and are usually measured in hours. Keep in mind that before meiosis begins at all, the DNA undergoes replication just like it did before mitosis started. So when you first see chromosomes in meiosis 1, they have sister chromatids, just like in mitosis. It is just that in meiosis 1, we will be talking about tetrads becoming visible, lining up, separating, and condensing, rather than chromosomes like in mitosis. Finally, cytokinesis occurs too, anytime after the tetrads have moved out of the equator, just like in mitosis. In prophase 1, just like in mitosis, during prophase, DNA condensation occurs. The nuclear envelope and nucleoli disappear, and the spindle starts to form. The big difference is what is going on the chromosomes themselves. As DNA condensation proceeds and the chromosomes first become visible, they are visible as tetrads, so tetrads become visible during prophase. Meiosis 1 begins with the condensation of the chromosomes in the leptotin during prophase 1. During the pachytene, it is the another stage of prophase 1. The pairs of homologous chromosomes align to form tetrads in the process called synapsis. Corresponding segments of DNA of sister chromatids of 2 homologous chromosomes twist and cross, forming so-called chiasmata. In these regions, exchange of DNA between homologous chromosomes can occur. Crossing over or crossover requires the chromosomes break and connect to the other chromosome. In metaphase 1, tetrads line up at the equator. The spindle has completely formed. It is during prophase 1 and metaphase 1 that genetic recombination is occurring. Keep in mind that it only happens when there are tetrads, so as soon as anaphase get going, genetic recombination is over. In anaphase 1, tetrads fall apart and chromosomes with 2 chromatids move over the poles. In telophase 1, chromosomes with 2 chromatids decondense and a nuclear envelope reforms around them. Each nucleus is now haploid. Keep in mind that it is not the number of chromatids per chromosome that determine whether a cell is diploid or haploid, but it is the number of chromosomes and whether they are paired that determines this. At the end of meiosis 1, each chromosome still had 2 chromatids, that is double the amount of DNA that a cell should have. So the entire reason to go through meiosis 2 is to reduce the amount of DNA back to normal. Basically, to split the chromosomes so that each daughter cell has only 1 chromatid per chromosome, the normal genetic content. You will see that it looks like mitosis. Keep in mind that the only difference is that the two chromatids per chromosome are not necessarily identical due to genetic recombination occurring in meiosis 1. And this will undergo meiosis 2. In prophase 2, chromosomes with two chromatids become visible as they condense, and the nuclear envelope and nuclei disappear and the spindle is forming. In metaphase 2, chromosomes with two chromatids line up at the equator, while the spindle is fully formed. Although genetic recombination primarily occurs during meiosis 1, the way the chromosomes line up during metaphase 2 can also help to make unique daughter cells. In anaphase 2, chromosomes split so that a chromosome with only one chromatid heads toward each pole. In telophase 2, chromosomes with only one chromatid condense and get surrounded by nuclear envelopes. The four daughter cells are now all haploid and have the right amount of DNA. They are ready to develop into sperm or new eggs now. And this simulation ends here. Thank you so much for watching.